Hello, everyone. Good morning. I'm gonna start by sharing my screen. Okay, the number seems steady. So welcome everyone and good morning. I'm Serena di Cecilia. You may know me if you have seen already some webinars from FlowJo. I'm the EMEA Software Solution Manager, and here on the screen you can find my email address and my colleague email address who is covering Europe together with me. So please feel free to email us in case you have any problem, any issue, any question that can arise during your analysis or while you're using FlowJo. There is a Q&A box that you can use for your questions. So today I'm alone. So I would kindly ask you to place your questions in the Q&A box so I can see that, and do not use the chat for the questions, because otherwise it gets complicated for me to double check that. I will stop or pause during this webinar to allow you to ask your questions once in a while, but please place your questions in the Q&A. I'll do my best to read them and to answer them live. Remember that this webinar is recorded, so in case you need to leave earlier or you can't follow through the end, this will be available on the website under recorded webinars in a couple of days max. Okay, so we held webinars monthly, so no worries if you can't follow this. There will be another one next month. And every month we held a couple of webinars, one for basic functions of FlowJo and one for advanced features of FlowJo, as well as for BD Research Cloud, which is our cloud environment for collaboration. Okay, so I think I may get started. So the first thing to know is that today this will be a webinar on FlowJo version 11. FlowJo version 11 is the new released version of the software. It's a modernized, updated version, and it was released back in May. And you can download it from the website as you downloaded FlowJo version 10. There are some differences in the software, but please know that if you are using a portal account or actually a subscription to FlowJo, you can use both versions and you can keep both versions on your computer. So you don't need to uninstall version 10 and install version 11. Please feel free to keep both on your computer to use both, because with subscriptions you can use both versions. One question that always comes up when we start talking about version 11 regards dongles. Dongles are our USB keys, and dongles do not support version 11. So in case you are using FlowJo via dongle, please feel free to start a free trial if you want to try version 11, and you will be given a portal account. Well, you will be given a temporary license, and you just need to create your portal account, which is basically based on username and password. And you can use this free trial for 30 days. You can analyze any kind of data. It's not just specific for demo data, but you can use your own data, and you can see if you like it or not. Let me try to open up the Q&A just to make sure. Okay, so there's nothing in the Q&A yet, but please, again, for those of you that just joined, feel free to ask your questions in the Q&A box. So when you land on the FlowJo webpage, you will find several tabs on top. And if you just navigate to the download area, you will be prompted to download FlowJo version 11. Here you can also ask for a free trial. There are several places where you can ask for free trial. Here on top of the right, corner or right here or down here. I see there are some Q&As coming up. So the first one is, will it be possible to convert the dongle license into an online license to be able to use FlowJo in the future? The answer is absolutely yes, depending on where you are located. I believe you are located at least on the east side of the world. So please contact your BD rep for that, or you can contact the FlowJo office as well. If you're based in Europe, contact your BD representative for that. And then there is another question. And if I want to continue with version 11, one has to buy a new dongle? I'm not sure I understood the question. If I want to continue with version 11, maybe you're referring to version 10. So the subscriptions cover both version 10 and version 11, while dongles only allow access to version 10. So I hope I have answered your question, but if I haven't, please feel free to clarify that. Okay. Okay, so the first thing to mention and to know when you start using version 11 is that we do have a new access system. Well, a new access system. It's actually similar to what we had with subscription in version 10, but now it's called BD Access Portal. So whenever you download the software, you will be downloading 2 applications this time, not only one. So you will find under the in the folder that you download, you will find the FlowJo application and the BD Access Portal application. So please download and install both. The download will be automatic, so you don't need to do anything. But for the installation, yes, you will need to approve the installation of both of them. If you are on a Mac, the installation is just drag and drop into the applications folder. If you are on a PC, the installation will be automatic. So you will just be prompted with some with a question like, would you like to install both? Yes, please say yes. Keep saying continue, and the installation will be automatic. Oh, okay. Now I see in the Q&A, if I understood it right, at the moment, there is no dongle option with version 11. No, that is correct. So once you have downloaded and installed both, the first thing to do in general, the first time you run FlowJo version 11, is to start BD Access Portal. This will automatically drive all the rest. Okay, so you will find it under your application or you will find it under your program files. Please double click on BD Access Portal to start it. You may not see anything on the screen, but the important thing is that it was started and launched. And then when you open up FlowJo, you will be prompted with this, which is your way to access the software. So you just insert your username and password and then just click sign in. Okay. You can note that on the bottom left corner, now this is a Wi-Fi sign, but we have changed it to a globe. The important thing to look out here is the color of that. So if it's green, it means that the connection works, everything is fine. If it's red, it means there is no internet connection. If it's yellow, it means you may have some IT issues, and please contact us at flowjo@bd.com in that case, and we can help you to troubleshoot everything. Okay, so just let's give a look at the actual, website, what we find on the website. So as I was mentioning, when you navigate to the FlowJo website, you will find several areas. There is the BD Research Cloud area, the FlowJo area, the FlowJo Exchange area, and so on. Note that if you want to use FlowJo, you can just click on the FlowJo page right here. And as I mentioned, you will be prompted to download FlowJo version 11. Start your free trial in case you don't have a portal subscription. You can ask for a free trial here. And the only thing that you need to do when you start a free trial is to send your information so you can actually, we can actually activate a temporary license for you. When you go on download, you will find several options depending on the machine that you're using. So the machine meaning the computer that you're using. So if you have an Intel Mac, or if you have an Apple Silicon Mac, or if you have a Windows. So please download the correct version. Otherwise, if you want to use FlowJo version 10, this is not the right webinar to follow, but you can download FlowJo version 10 here. There are many webinars on FlowJo version 10. So in case you want to use the previous version, please feel free to see and rewatch the recordings of that. I see I have a couple of more questions. Is the recording of today's session will be available? Yes. BD Access portal is not working. I can connect on the FlowJo portal and not on the BD Access portal. So in this case, please send an email to flowjo@bd.com, and I will be happy to troubleshoot and to help you right away. Well, this afternoon if you're based in the east side. Is it possible to work offline, or does the BD Access portal need to be on all the time? No, you can also work offline, and you can also request a token for 2 weeks. So all these information are available on the FlowJo documentation, which I'm going to show you in a second. Okay, so where do I find the FlowJo documentation, the webinars, and everything? So if you go under Learn, right here, you will find 4 different tabs. One is related to webinars. So you may have seen that if you are in this webinar today. So here there are all the upcoming webinars. There is another introduction to V11 in October from John Quinn, but we also do have webinars regarding imaging analysis and BD Research Cloud. You can register to these ones. And then this webinar, you will find it right here under all previously recorded webinars. So there was one as well in July that I did, which is already available here. And you can navigate and find all the many webinars that we have. The second thing that I want to mention is the FlowJo University. The FlowJo University is a repository of short videos. So in case you don't have time to watch a full webinar of an hour, please feel free to check the FlowJo University right here, where we have short videos on how to perform actions or how to create something in both version 11 or version 10. So if you go under FlowJo version 11, okay, start here. You will find all the information on how to download and install FlowJo version 11. You will see there are 3 videos, one for Windows, one for Mac, how to sign up for a free trial. So all the information are there. And then you will find as well videos on how to perform things in version 11. The Metadata Manager, the quality control, compensation, graph platforms. And as you see, these are like really short videos, 5 minutes to 10 minutes videos. So you can get right away to what you're looking for. So please feel free to check these videos. And then finally, what I was mentioning before is the help documentation. So if you navigate to the documentation of FlowJo version 11 and you go on read documentation, right here you have FlowJo version 11 licensing information. So how to install, how to update, and so on. So for example, in the installation, as I was mentioning before, there are all the screenshots for PC, for Mac, everything you need to know, it's right here. Okay, so let me pause for a second. This is a question that I will ask in a second, Mark. Okay. Okay, so we will cover that when we get there. Okay, so if there are no other questions regarding the installation and the download of FlowJo version 11, let's dive right into the software. As you can see, I have BD Access Portal right here on top of my screen because I launched it before, and now I'm gonna just open my FlowJo version 11. So now I have a dark theme. This is actually my dark theme. It's called Serena Dark, but you can change the theme. So that's what I'm going to do now. I'm going to change it to a light theme. So if you go under view right here, you will find different themes. So you have a light theme, chorus theme, dark, and so many other themes. So let's just go with the light theme, which is the classical one. And then when you open up this workbench, now we don't call it anymore workspace, we call it workbench. You will have all the links to the FlowJo website, to BD Research Cloud, to the documentation, and to FlowJo University. So it's really easy to access all this information at this time. You don't need to have your web page, a separate web page open, but you can just navigate there from here. You will also find on the left different actions. So you can open up a new workbench or a previous one, one of the ones that you have created, but you will also find a list of all your previous workbenches right here. So these can, let's say, facilitate the finding of your last workbench or your previous workbench that you want to open. So let's start with a new workbench. So I'm just going to click on new workbench, and I had another one on the background that I was running. So I will just say yes, save it on my desktop. Okay, and now I'm opening up a new workbench. So the first thing that you notice is that now we only have a single window. So there is just this workbench. There are no other individual windows running around or opening up and closing and so on. And this workbench is mirroring, let's say, your bench in the lab. So we have all the samples on the left side, our work in the center, and then on the right, let's say we have all our reagents. So basically we have all the characteristics, all the settings for what we are doing. And you will also notice that we don't have a horizontal bar on top anymore, but all the activities are or can be accessed by the left side of the panel right here, the navigation bar. We have tried to create a workflow for you or help you to follow a workflow during your analysis. So ideally, the first thing that you would do would be Metadata Manager. So meaning that you want to name your files or label your files. You want to add extra informations. You want to modify your parameter names or stain names and so on. Then we will have a quality check that you can choose to run or not. That's up to you, but I would recommend you to do that. And then we have the compensation platform right here, where you can either create a new compensation matrix or edit a previous compensation matrix and so on. The graphs are right here, the graph platforms. And then we have something called platforms. So now we have included some high dimensional analysis algorithms in FlowJo version 11. And we hope to have new algorithms coming soon in the next updates. For now, we have t-SNE for dimensionality reduction and XShift for clustering. You will then want to create a table for your data. So right here under tables, and then the ability to create charts starting from that specific table. So you may remember if you have used FlowJo previously that if you want to create a chart, you have to first create a table and then export that into something that can allow you to create a chart, such as, for example, Excel. In this case, we don't have to do that anymore. We have charts right here, which will be built based on the table that we have. And then finally, our layout editor becomes the report editor. So now we can create all our reports right here. In terms of adding samples, pretty much similar to version 10, we can drag and drop samples, or we can click on the tube icon right here, where we can select which samples do we want from a location. So if I click here, add samples, I have something on my desktop. This should be my data, basic training. I can open. And now I have all my data right here. First of all, let's clarify a second which data I have. If you have seen any previous webinar, these are exactly the same files. So we're using the same training files. This is a phosphoflow and intracellular cytokine staining where we thaw and rest cryopreserved human PBMCs overnight, stimulate with PMA plus inomycin for 2 hours, and then stain for viability. So there is a first surface staining for viability and surface antigens, and then another stimulation for 20 minutes with PI, and then we fix and permeabilize the cells to allow intracellular staining. So there are 2 different stainings, one for surface antigens and one for intracellular antigens, and then 2 different stimulations. So we basically have 4 potential combinations for each sample, and in this case I have only 2 donors, but overall the data set that I have includes stained samples, includes FMOs, which are our controls, fluorescence -1, where we have everything in the tube except for the marker that we want to check, and then we have compensation controls based on cells and beads. So the first thing that I see is that now, let's say what we used to call our groups, well, our all samples group is our acquired data right here. And under here, I have my compensation data, my tubes. So the idea is that we can have different compensation sets of samples. So we can create groups under the compensation data. Now I have one set of compensation controls, which is right here under my compensation data. And then we have all our compensation matrices. In this case, I only have my acquisition defined matrix, but in case I calculate a new matrix or I modify or copy or duplicate a matrix, I will find them under here. And then I have my experiment data, which are actually the stained samples. In this case, I have everything into a single group. So everything that I had in my folder is under basic training. I can, of course, edit the name of this group. So as we were doing in FlowJo version 10, I can right click and edit. Maybe I can call this group donors. Okay, and you will see that you can increase and decrease the size of this. You can close it if you want to have more space and so on. So since this is my donors group, I can remove my FMOs from this group. And now I only have 8 samples. These are 2 donors, 4 different stimulation conditions. But I still have 27 samples in all my acquired data. So now I can create another group for my FMOs if I want. So I can right click, create child group. Remember that you can also create a group starting from this icon right here. These are pretty similar to the V10 icons that we have. This is adding a sample, creating a group, add statistics right here in the samples area, and add keywords in the samples area. So let me create a child group. These will be my FMOs. And as we were doing before, we can use the file name to allow FlowJo to include in this group all the files that in their name contains the word FMO. And here it is. Now I have 2 groups, one of donors and one of my FMOs. And the first thing that you see is that now I have the visual on all my samples at once. So I can scroll down and see my main sample right here in the main graph, but if in the graph gallery, I have the ability to look at all my different samples. I see there is a question. Regarding the compensation, maybe I missed it, but did you mention the possibility to have different groups of single stains and then choose the one we want to recalculate the compensation matrix? Yes, and you can do that in V10 as well. Let's say you run you run your experiment, you run a set of compensation controls one day, and then you realize something was wrong with your compensation controls, you want to rerun your compensation controls, remake them, you can do that. You can have two different groups for compensation, you can calculate two different matrices. Okay, so now I see all my samples down here. This gives me right away a visual of everything that is happening within my data. And then if I move under experiment data, now I see all my FMOs and all my donors. So probably one of the things that I want to do is, first of all, evaluate my information on the data. So if we start from the first tab on top, this is Metadata Manager. What do we do with the Metadata Manager? We have all the informations related to the sample name and all the keywords associated to our file, even from the instrument. So sometimes we don't even know that we have all these information associated to the file, but these are actually some really helpful informations that we get that the instrument itself applies to the samples. So for example, we find the date of acquisition, we find a keyword if we added that on the instrument, the CSNT beats lots, and so on. So all these informations are listed here, and you see different keywords columns for each sample, but if you see this visual is not convenient for you to look for your specific keyword, you can also pivot this table. And now I have each column that is a sample rather than a keyword. And then all my rows are my keywords. So now I can scroll down, look for a specific sample, and check all the keywords for that sample. So up to you the way you prefer to look at your sample. Okay. Okay, gonna add answer in a second. If you're looking for something, you can just filter keywords, PMTs. So there is a question in the chat that says, in regards to the metadata, I'm not able to find the PMT used. Can you show how to find it? It's possible that has a different keyword name. So my advice, I can't find it either in this case. So my advice would be to either go back to V10 and see which is the actual keyword that you have. So usually there is a $sign or something like that next to it. But what you can do in that case, you can also have information regarding all your parameters and parameters names. If you move on to the parameters tab right here, and then in this case, what we can do, sometimes what happens is that we made a typo in our stains. So please remember that parameter name and parameter stain are two different things. Parameter name is actually what we use, like PE or PE Sci5 or FITSI and so on. Parameter stain is what we assign to each of these parameters. So if we assign a staining name as CD3, CD4, CD8, and so on. So you can edit that. If you realize that you made a typo, you can just click on the pen right here and then edit your parameter, your stain name. And then finally, we can create parameter sets. The idea of creating parameter sets is linked more to a need of shortening the list of parameters that we have. And this is really useful in case we have a long list of parameters. For example, we're working with 30 colors, we use our lineage markers, and then we use our stimulation and so on. And so once we have cleaned up our gating from the lineage markers, we may say that we don't want to have these markers in the list anymore. So we want to make a list shorter to filter out the parameters faster. So for example, I could also create here just one list that includes phospho-ERK, perforin, and interferon gamma, and call this one my intracellular, for example. Okay, so now I have a new parameter set, and this only includes 3 different markers that are my intracellular markers. Let me just give a look to the Q&A. Okay. Is it possible to zoom in on a single graph without zooming in on the whole screen? It haven't been able to do that, and it can be tricky. No, that's the answer. You cannot zoom in into a single graph. We are working on high graph resolution, so will come. Just be patient. Okay, and then do you go through the compensation? Is it possible to use peak instead of area for compensation? Let's see if we can cover that while we go through the webinar, because this is an introduction to FlowJo. There will be a compensation webinar specific for that. So let's see if we can cover that. And then the last question I have is, I may have missed it, can FlowJo v11 open workspaces from older FlowJo versions? Not this version. So you cannot open a v10 workspace into v11 at the moment, but it will come. So this is the first priority, the first request that we have. So working on it, just give us a little time. And we release frequent updates of FlowJo, so just be look out for it. Okay, so now we have gone through the Metadata Manager. In case you want to add a new keyword, just click on the keyword tab right here, and then you can assign a name, and you will have your prompt to add the keyword. Okay, so the next step would be a quality check. So whenever you move on to the quality check, you see it's empty because FlowJo is saying choose the parameters and adjust the settings to apply the quality control to the experiment. You don't have to adjust the settings if it's not needed, but what you have to do, or maybe double check, is that it is running on the parameters that you want to check. So I usually don't check physical parameters because I'm looking at fluorescence anomalies. So in this case, I deselect my physical parameters and I run it on my fluorescent parameters. And this quality check is FlowAI based, which means that it makes 3 different checks. It checks the signal stability, the flow rate, and the dynamic range of the instrument. So basically, how stable was the signal over the acquisition? Did we have increase of the signal or decreases of the signal? Was the flow rate stable during the acquisition? Do we have a drop of the flow rate? Do we have a spike of the flow rate? Do we have clogs and stuff like that? And finally, do we have events that are falling outside of the dynamic range of the instrument? So basically having values that are really, really low or really, really high, which are actually outliers. You can choose to perform all the different checks or just one of them. I did release there is a webinar on quality check that goes deeply through all these characteristics right here, but even in the documentation, you will find all the different parameters, how you can adjust it and basically make it less stringent or more stringent. For now, we are just going to apply the default parameters, see what happens. Okay, and now what do I have is my samples cleaned up. And basically here I see one parameter versus time. This is my AARD, my dead parameter versus time. Here I have my flow rate and where I got the drop of the flow rate. And then down here I have the signal stability over time for all the parameters. If you want to look at one specific parameter, you can also just navigate to that and you can scroll down to your samples and make sure that everything is fine. Okay, now talking about quality check, there were a couple of questions in the chat. One is, after performing QC of the unmixed FCS files, do I have to also proceed with the time gate? No, because what you're doing with the QC is automatically excluding these areas. So with the time gate, what we would do would be manually creating a gate around this to exclude this area. And then we go here, exclude this area, and then we close the gate. But that's actually what the software is doing. So you don't need to create a time gate. And indeed, your following question is, because when you try creating one, the populations are disjoint. And of course, you don't have to create a time gate once you do the quality check. Okay, I hope I answered your question. Now what we see is two new populations that have been created, one on bad events and one on good events. Again, the bad events will be all these events right here where the where the interval of time was excluded, basically, and the good events are all the rest of the events. I can scroll down and see my different samples. Okay, and I can move around, evaluate, and so on. So when you look at the statistics, now we have if I click on the experiment data, so please make sure you are on the right either group or population to look at your data, because now I'm looking at all the different events coming from my experiment data. And when I'm starting to gate, I will start from my good events. Okay, so in case of the compensation, if you have compensation controls, and again, I don't think we have time today to go through the compensation, but if you do have compensation controls, you can just go and click next, and we'll automatically start creating your preview. Okay, so basically, as we had in V10, you will have a list of your controls, and then you have a green dot if it's paired correctly. You have a rainbow if this is used as a stained sample, or you can uncheck it if you want to use it as an unstained. And then you will have auto spill by default set to be on. Auto spill on means that I don't have to create gates on negative and positive peaks. So there is also a short video on compensation in the FlowJo University of 10 minutes. So please double check that if you want to know more about that. So if I go with traditional compensation, I have a preview. So what it does, it's basically calculating a preview of my matrix, and then I can create the matrix. So in this case, I can scroll down and look at my parameter versus the rest of the parameters. So we don't have anymore the n by n, so each parameter versus the other, but we have one parameter at the time. I can scroll down and look at whatever I need to look at. I can use a dot plot or a contour to look at my compensation, and then on top we have our matrix. We can you can also increase the size of these in case you need. And if you don't have enough space, you can always close your side wings. You will also be able to create a compensation correction in case of post unmixing, in case you want to create post unmixing correction, and you will have a spillover spread matrix or a total spread matrix, depending on what you're working on. And then you can create matrix. Now my new matrix as a different color, as you see here, is under my compensation matrices group. And I can, as I was doing for my V10, I can just move on to my experiment data, and I would or could eventually look at my compensation on my data. The best thing here, I think one of the coolest features, is that if you navigate on one of your samples, whatever you are, okay, When you are in the compensation settings right here, if you see something wrong and you select the plot, automatically highlights which is the combination. And this, at least for me, has been quite a challenge in the past to, look at what is what and what should I look at. So now if you just navigate through that, highlights automatically the plot. Same thing if you select anything on the matrix, it will automatically update underneath. Okay. When trying the compensation wizard, our parameters were not 100% when compared to the same parameter. I'm not sure I understand this question. Can you please rephrase it? Okay, so then I can take my matrix and apply it to my samples. In this case, I don't really need to do that, as this is exactly the same matrix as my acquisition defined. At the moment, we can't compare two matrices as we used to do in B10, but this as well is a feature request, so it's on the list. We have about 400 to 500 feature requests, so let just give us the time to work through that. And finally, we are at our gating strategy. So I don't want to spend too much time on the compensation. First, because there will be a webinar specific on that. And second, because we have a video on compensation on the FlowJo University. And third, because not everyone that is on this webinar is actually interested in doing the compensation. Most of the people do the compensation on the instrument, so they don't recalculate that in FlowJo. So I want to be happy to make everyone happy, actually, and go through a full workflow as much as I can. Okay, so now on the experiment data, we can start gating. Again, the reason why I told you, can you just make sure the group you are on, because if I'm now, one of the changes that we have is the gating hierarchy. So if I'm on the experiment data, whatever gate I create right here, it will be automatically applied to the rest of my samples. So if I'm on the experiment data, I start from my good events, and now what I can do is start creating my gates, because basically I want to create the same gate in both my donors and my FMOs, at least until now. So I can just choose an ellipse gate. On the top, you find all the tools to create a gate. Yes, lymphocytes is fine for me, but you can assign a different name as well. And then you see that now all my samples have the same gating strategy, or the same gate applied. What is the graph gallery and how does the graph gallery works? So here you find two options. One is graph gallery, one is image wall. If you don't have image data, then the graph gallery is the only thing that you can use. If you have imaging data coming from our Discover instrument, you can use as well the image wall, and here is where you will see the imaging. And then you can choose your visualization way. So you can look at it by sample or by parameter or by ancestry. So let's start by looking at it by samples. Then by the time that we do more gating, we will look at the different ways. So to create the second gate, now I have my gate right here, my lymphocytes. I can go through all of my samples, and within my, if I want to go back to my parental gate, again, I have my up and down arrows right here, and then I can move on and create a second gate onto my single set. Okay. So I can exclude doublets. I can use a polygon gate for that. Okay. If you double click, the gate will close automatically. I can call this single cells or singlets. And then within my singlets, I can look at my live cells. And now what we start seeing is that we have our axis or our data that would need a little bit of transformation. So in this case, my population is really squished. I want to make it a little bit open. And so I can show you how to transform your axis. Again, we have T buttons to transform the axis. So I can click on the T right here, and my parameter list will be right here. In this case, you can as well choose a parameter set that you want to transform, and you will see now here my intracellular parameter set, scatter sets, compensated, uncompensated, image derived, and so on. I can just keep it to all for now because I just want to change my AARD, and I just want to make my cofactor, change my cofactor, and actually change the width basis a little bit. Apply. Here we go. Now it is applied to all of my samples, and I can create my gate on my live cells. And then within my live cells, I can look at my DR versus CD3. And even in this case, here it's really clear that I need to transform my axis at this point. I want my data to be correctly displayed. And just remember that transforming the axis is not changing your data, it's just changing the way your data is shown. So I want to see the actual separation between negative and positive. I want to make my cells a little bit larger, my populations a little bit more resolved. So I can, even in this case, change the width basis. I have too much white space. I don't need it because I don't have these values so high. So I can remove some positive decades from here just by clicking on the minus. And then if I click apply, it starts looking a little bit better, but I still need to, let's say, kind of transform my DR. So I can do the same thing here. Okay, now looks much better, let's say. I can still remove one of the positive decades. That's up to you how you prefer to look at your data, but I personally prefer to look at it this way. So now I can gate on my CD3 cells, and you can choose all the different ways of gating. Sorry if I go by rectangle gate, but it's automatic by now. But you can choose a pencil gate, okay, like this one, or you can choose to use the quad gate or the curly or the spider, the same as we did have in V10. Okay, now I finish my gating and I look at the Q&As. So the last gate that I want to create is CD4 versus CD8. Okay. Even in this case, I totally need to transform my axis because I can't really see my cells, right? So I'm gonna do something like that. Okay. And probably even here, I can remove. Maybe that's too much. Here we go. So this would be my CD4 cells. And this would be my CD8 cells. Okay, so let's look at the Q&A if there's something that I can enter. Okay, if I have made a square gate but wants to change it to a polygon, it was possible to do it in V10, will also come in V11. So we're trying to include as many features as possible. V10 has been out for at least 10 years, so we do need some time. Okay. Okay. So in this case, when we look at a graph or we create a gate, we only see one sample at the time. But there is something new in V11, which is the virtual concatenation. So basically, if I click this tube up here, I'm able to see all my samples at the same time on a single plot. This is something that in version 10 needed to be done manually. So I had to go to a concatenation step to look at all my samples at the same time. Now it's virtually performed, so you can create that automatically by clicking on these either 3 tubes or going back to a single sample this way. What is the advantage in this case is that I can easily create a gate, make sure that my gate is actually including all my samples or the events in all my samples. I could eventually make this one a little bit bigger, for example, to make sure that I include everything, okay? How do I change what I'm looking at on my plot? Well, on the right side, you have all the properties. So I can select the first icon, this one right here with 3 dots. This allows you to choose the plot type. This can be a pseudo color, contour, or histogram, or dot plot, and you can change the color, you can smooth, and so on. You can also choose different palettes, okay? So instead of using FlowJo Classic, I can use Viridis if I'm color blinded, or I can use Inferno or Blues, you know, that's up to you. Which one do you prefer? I'm just going with the classic one. On the X and Y axis, the icon that you see here, you can choose what are the parameters that you are looking at and that are displayed on your axis. So here is where it comes handy to have a parameter set, because in that case, for example, I can also just look at my intracellular markers and I have only 3 parameters here, so it gets much, much easier and faster to analyze my data. Okay. Okay. Where were we here? Okay. Where are my, this is my CD4, CD8. Okay. Here we go. And then finally on the T, you can transform your axis. The last icon is related to the image wall. So again, in case you have images, here is where you will find your image data, and underneath here, you see your image wall. So instead of looking at the image wall, we want the graph gallery. And what else we can do here? So we can look at our samples one by one, so by sample, or we can look at it by parameter, which means that I can choose I can choose to look at to have one parameter fixed either on my X or Y axis. So now it's batching everything on the X axis, which means that my parameter is fixed on the Y axis. I have CD4 versus everything else. And from here, I can realize where I have problems, if I do have problems, if I need to change my compensation, and so on. And finally, you will have a view by ancestry, which is actually showing you the full gating strategy that you did to get to this point. So these are my good events, physical parameters, singlets, live, CD3, CD4, CD8. Okay. The next icon is the advanced platforms, which we will not use today because it's high dimensional analysis. So this is mostly an introduction to FlowJo. So the idea is to give a webinar for people that are starting to use FlowJo or are starting flow cytometry. So I don't want to be too much specific on these or go too deep dive. Again, if you do have any specific question, please contact flowjo@bd.com. This is our technical support. They're super happy to help you. You can have a screen share with them. So please feel free to contact them. The next step would be the tables. So just one thing I want to do is creating my interferon gamma and perforin gates at least. So I can go onto my FMO group. I can do in this case two things. I can go onto my experiment data, look at the FMO that does not contain interferon gamma, which is this one. Look at my CD8 versus interferon gamma. Okay, which is this one right here. I can make a little bit of transformation of my axis and create my gate. This is my interferon gamma gate, FMO gate, okay. And now if I look at my actual sample, I see that there is a production of interferon gamma. If I move on my donors group, these are just the full stained, and these are my LD1 and LD2, two donors, two conditions of stimulation. So condition one, unstimulated versus non-stimulated, non-stimulated plus PI plus non-stimulated PI plus PI. So I can see how the amount of interferon gamma is increasing, and I can do the same by navigating onto my next sample. Okay, so I hope this is clear. I can do the same for perforin and phosphor-ERK, but I don't want to take too much time. I think we have almost reached the top of the hour, but I want to show you how to create a chart and how to create a table. So if you move on to the next tab right here, this is a table tab. And what you need to do here is first dragging and dropping samples or groups to generate the rows, and then populations and statistics to generate the columns. So if I want to have my statistics related to my donors, I can just drag and drop my donors group. And then what else do I want? I probably want the frequency of parent of all these statistics right here, up to you, or just of these. Okay, so you'll see by default we'll add frequency of parent, but you can add any statistics that you want. So let's say I want to add the median within my CD8. So I want to have my MFI. So you will select the population. You have it also here. Median of interferon gamma. Add statistics. Now I have this value right here, and I can add keywords as well to my table. So if you want to create a nice chart, I strongly recommend you to use keywords within your tables. So I can add keyword. This can be my, I think I have one condition keyword. Yes, add keyword. Now I have my keyword right here. I can order these ones based on, alphabetically, or I can order them based on condition, up to you. Which one do you prefer? Once your table is ready, you can either create a new table clicking on the plus button, duplicate this one, or remove it, or you can export it, and you can include or not the summary statistics. Is it possible to create Boolean gates? Yes, you select you go under your create Boolean gates. Okay, let me see. Okay. So once I have my table created right here, you can assign a different name if you want. You can then go on to charts. And don't be surprised if when you open up your chart tab, you don't see anything. Because FlowJo is telling you choose a source table that contains keywords, select category and numeric factors, and adjust the style then. So I have my table right here. I have my keywords. If you don't have any keyword, it will use the file name. But if you do have a keyword, which I recommend you to do, it will show you the keyword right here. And then you can select the specific keyword that you want to display. Let's say I want to show all my keyword and then, or basically all my different conditions, and then which statistic do I want to show? It can be my frequency of parent, frequency of CD4, frequency of CD3, frequency of interferon gamma, and so on. Okay, so once you have created your chart, you can choose, you have underneath the list of all your data, and you see these are adjusted, no, I don't remember how to say that, but it's basically rounded up, okay, rounded up to the closest number in your table. But then you have, of course, the in your legend. But then you have, of course, all the statistics in your table. You can change the color if you want. If you move on to the style, this can be a bar chart or can be a pie chart, the statistics to show. And then you can choose a palette. So you can use green blues, or you can use sign bow. Up to you, which one do you prefer? So let's go back to the bar chart, because I also want to show you what else you can do. So under the third icon right here, you can actually, again, change what you want to see. If you don't want to see one condition, for example, you can just remove it from the chart and just compare two different conditions instead of four. Okay, that's up to you. What do you prefer? And finally, you can add statistics. So you can choose a statistical test. For example, I can choose a Wilcoxon test, which is my baseline for comparison, is condition 1. And then which values do I want to compare? So I want to compare condition 1 with condition 2, 3, and 4, or just with 2 and 3, and so on. You can choose a threshold for significance. You can add a second threshold. And then finally, you can show asterisks for significance. You can show connecting lines, and you can show the p-value as well. Okay, now I have my p-values. Nothing is statistically significant. I don't have asterisks. Once I'm done with my chart, I'm happy. I can move on to my report editor. Well, you can also go directly to the report editor if you don't want to create tables or charts. But the last thing that I want to show you is how to create a report within the report editor. So you will just need to select your population that you want to see. Okay, so for example, I can drag and drop my singlets right here, and this is zoomed out. So if you click on this arrow icon, it is zoomed, that was zoom in, this is zoomed out. So I can actually move on to my next gating, CD3, and then my CD4, CD8, up to you. Okay, let's do CD8. Okay, here it is. So now I have my 4 plots. I can, of course, increase this. You can make it the size that you prefer. Now that I have all my samples here, I can use this small hand to move around my, my report. Okay. You can choose an orientation, so it can be landscape, portrait, PowerPoint, custom. Okay. In my case, it's custom. You can choose the size as well. You can export as one page, and you can export as a PDF. And once you have your, let's say, your data defined, and this may be your initial report, and you want to create the same thing for all of your samples, you can batch that. You can batch all the samples on a single page, or let's do first view batch. So now each sample is on a single page. Even if you don't see that here, underneath there are several pages now that I can navigate through. And these are relative to each individual sample. But probably this is not really nice. I want to have multiple samples. Maybe I only need 2 pages. So if I go back to view source and I use 4 rows, okay, so I have 4 conditions for each sample, I can do that. And now I have sample 1 and sample 2 right here. One more thing that you can do when you create a report is adding charts. So you can add your chart to the report. And again, you can zoom in, zoom out. You can add your chart as well as you can add a table as well in here. Let me see if there are questions here. Is it possible to export SVG files like FlowJo v11? Not in this version, but it's coming in next. Thank you. Same thing for Enrico. Can you also export in other format? Not in v11.0, but coming in the next version. Okay. Is it possible to have 2 workspaces open at the same time? No, but you can go back to another workspace just by clicking on the FlowJo icon here. Not for now. Okay. Okay, so any other question that may arise? I know we are 7 minutes over the time of the webinar. Please remember, if you do have any question, let me type it in the chat right here. FlowJo@bd.com. They will be happy and they will be the best to answer all of your questions. Okay. Can you iterate through samples in the same plot window? Kevin, I'm not sure I understood your question, but you do have several options for iterations right here, batch iteration. So you can iterate by population if you want. You can iterate by keywords, by statistics. Well, you can batch by population if you want. So depending on, I'm not sure I got your question. And then there is someone that is asking how to install plugins. There are no plugins. So no need at this point of plugins anymore. If you're using any plugin which is not present, any algorithm that is not present in v11, one thing that you can do is save, oops. Okay, sorry. Is save your workspace as, why I can't type. Save your workspace as a v10 compatible. So you can go under File and you can export as a FlowJo v10 workspace. So if you feel you're using some algorithms in v10 which are not present in v11, you can always do that. That's an interesting question. Hi, Serena. In the charts for the condition keyword, can you select the order 2413 instead of 1234? When I have wild type versus other condition, wild type is always at the end. That's an interesting question, but I believe my suggestion here would be to add the keyword just with the order that you prefer. So assign a keyword that can be order, for example, and name your files, name the keyword values with 1, 2, 3, 4, the order that you want to display them. So that will help you to display it in the right way. Can we export an FCS file with compensation matrices, for example? There is no export of files yet in V11. All these things are coming because I guarantee you 90% of the questions that I got this morning are already in future requests, are already in development, if not already developed. Then I have another question. The Boolean created in V11 didn't work in V10. I was talking about saving as V10 compatible file. Maybe if you can share the data with us, that would be really, really helpful. For those of you for creating the Boolean gates into V11 and exporting in V10, I would be really, really curious of seeing that. Okay. And then, is the webinar recorded? Yes, it is recorded. You should be receiving an email with the recording link. If you don't receive the email, just go on the FlowJo webinar, on the FlowJo webpage, and you will find all the recorded webinars there in a couple of days. Okay, I think I need to drop. We have been 11 minutes away. For the questions that were not answered, please double check with the FlowJo tech support team at flowjo@bd.com. If I can, I will be happy to help you as well. The last question that I see is, can data output by a spectrum analyzer be analyzed with version 11? Totally. It's amazing to work with spectral data in V11 because you can do the unmixing, you can do the post-unmixing correction. It's great. Yes, totally. Okay, so I think it's time to wrap up. Thank you so much for joining. If you do have any question, again, please reach out. And I'll see you next month. Actually, there is an advanced webinar on version 10. Bye-bye.